Overall, the steps and stages of meiosis are the same as the steps and stages of mitosis. So there's a prophase where all the same things happen. The nuclear envelope breaks down, the DNA condenses, the mitotic spindle grows. In metaphase, the chromosomes line up along the metaphase plate. Anaphase, we have separation of chromosomes. Telophase, the nuclear envelope starts to reform, the chromosomes decondense, mitotic spindle shrinks, and cytokinesis forms. And then again, the same basic steps happen during meiosis too. However, there are differences between meiosis versus mitosis, and pretty much all of them happen during meiosis 1. So just a quick list of them, and we're going to go through each of them. The differences between mitosis versus meiosis 1 is there are several things that happen during meiosis 1 that don't happen during mitosis. And those are pairing and synapsis, crossing over, and separation of homologous chromosomes. So the first thing that happens is pairing and synapsis. So here's our cell in early prophase of meiosis 1. The DNA is starting to to condense. This is during the mitotic phase, so DNA replication already happened during S phase of interphase. And the first thing that has to happen is the homologous chromosomes have to find each other. So they're floating around in the cell and they have to line up and match and find their homologous partner. So if the wrong ones match up, okay, there are proteins that actually read the nucleotide sequence and say, wait, these are not a match, and they don't attach to one another. If there is a match, okay, then the synaptonemal complex will hold them together. So the synaptonemal complex is a series of proteins that are going to hold the homologous chromosomes together during prophase of meiosis 1. So there's actually a reading mechanism that checks to make sure that the nucleotide sequence on the homologous pairs is a close match. They're not identical, right? Because our homologous chromosomes from mom and dad could have different alleles or different versions of the genes on them. So they're not identical, but they have close sequence homology. They're very similar. And if there's a match, this protein structure called the synaptonemal complex basically stitches these homologous chromosomes together. It's almost like a zipper holding them together. So one thing that's unique to meiosis that doesn't happen during mitosis is pairing and synapsis of homologous chromosomes. Next thing that happens that's unique to meiosis also happens during prophase of meiosis 1, and that's crossing over. So crossing over happens between non-sister chromatids of homologous chromosomes. So pairing has to happen first to make sure that they're matched up with their partner. Okay? And the crossover happens between one chromatid from one chromosome and one chromatid from the other chromosome. It doesn't make sense for sister chromatids to cross over because they're identical, right? So crossing over has to happen between non-sister chromatids of homologous chromosomes. Crossing over is going to happen on every pair of homologous chromosomes. It also happens on the sex chromosomes, on the X and the Y. There's a small region, even though those two chromosomes are very genetically different. The tips of the chromosomes have this region called the pseudo-autosomal region. And at that point, they have sequence homology and can cross over, even though all the rest of the chromosome has completely different genes between the X chromosome and the Y chromosome. So the purpose of crossing over is actually to double check and make sure that the things that are paired up are homologous chromosomes. If they're too dissimilar, crossing over can't happen. So the purpose of crossing over is to confirm that the right chromosomes are paired and synapsed, that homologous chromosomes are together. But we're going to talk in a little bit about how there's the additional benefit of increasing genetic variation through crossing over. So the last thing that's unique to meiosis is what separates during meiosis 1. So we go to metaphase, and these chromosomes are going to line up along the metaphase plate. We'll turn it like this, okay? And then during mitosis, it would look like this, right? During mitosis, nothing would be paired up. During mitosis, we would separate sister chromatids. But since we did pairing and synapsis and crossover during meiosis 1, these homologous chromosomes are together during meiosis 1. And what we're going to separate during meiosis 1 are those homologous chromosomes. And if crossing over has happened, the chromosomes that are separated from one another will be made of sister chromatids that are no longer identical because of crossing over. So the third thing that's different about meiosis versus mitosis is that in meiosis, it's the homologous chromosomes that separate at the end of meiosis 1. Then as we go into meiosis 2, so we're going to move this cell off the screen. Now we have just one cell here. This cell has just one of each chromosome, but every chromosome is made of 2 chromatids. Okay? So in this cell, there's 2 chromosomes, one long one and one short one. Even though this is made of 2 pieces, this is one whole chromosome. Okay? But we have 2 copies of every gene. So we would say that this cell is haploid because it has only one of each chromosome. But it's also 2N because it has 2 copies of every gene, okay, one on each chromatid. And then in meiosis 2, what we separate in meiosis 2 are the sister chromatids. But those sister chromatids might not be identical anymore because of crossing over. Okay. So when this happens, we end up with, at the end of meiosis, gametes that are haploid. They have just one long and one short chromosome. But now they're just 1N. They have just one copy of every gene. And so at fertilization, when we get another gamete that has one of each chromosome, we get back to our cell, our single-celled zygote, that is both diploid and 2N. The big differences between mitosis versus meiosis is the genetic variation in the cells that are produced. In mitosis, we go from diploid cells that the original cell is diploid and it produces 2 daughter cells that are also diploid and genetically identical to the parent cell. But in meiosis, the whole purpose of meiosis is to cut the chromosome number in half, to convert a diploid cell into haploid cells. And in this process, we end up with cells that are genetically distinct from one another. They're different from the parent cell, but they're also different from each other. And so there's a couple of things that happen during meiosis that lead to genetic variation. So when we talk about genetic variation, when we talk about genetics, we're studying about the heredity and variation from one generation to the next. So heredity is heritability, right? Transmitting traits from one generation to the next. And variation is differences in appearance. So having dark hair or light hair or freckles or no freckles or widow's peak or not a widow's peak. Okay. So the raw material for variation comes from mutations. Sexual reproduction alone doesn't create new genes. All it does is mixes them together in new combinations. The source of new genes are mutations. And if you get a mutation in an existing gene, you create a new allele. So if we think about the arrangement of genes on chromosomes, in a diploid organism, you have a homologous pair of chromosomes, one that came from mom and one that came from dad. And at a particular spot or locus, okay, you can have different versions of that gene. You can have 2 of the same version of that gene. You could have 2 different versions of those genes. And the different versions of those genes are called alleles. What sexual reproduction can do is mix different alleles together in new combinations in the offspring that result in a unique combination of phenotypes. And there's 3 different ways that this happens: independent assortment, crossing over, and random fertilization. And we're going to go through each of them. The one we're going to talk about is independent assortment. Independent assortment has to do with how the chromosomes line up at the metaphase plate during meiosis 1. Okay. So in our simple model organism here that has 2 different kinds of chromosomes, we're going to assume that the chromosomes from one parent contain the recessive alleles and the chromosomes from the other parent contain the dominant alleles. Now it's possible that the chromosomes could line up like this along the metaphase plate. And so if we go through meiosis 1, okay, all the little a's end up in one, the lowercase alleles end up in one cell, and the large dominant alleles end up in the other cell. And then if we finish meiosis 2, we'll end up with two gametes that are the same that both have dominance, and 2 gametes that are both the same that both have the recessive alleles. So we have yellow with blue and red with green. But that's not the only way they could have lined up on the metaphase plate. We could have lined them up like this, so that we had big B up here and little b down here. And now when we go through meiosis, we'll end up with gametes that have a different genetic combination. We'll have little a with big B and big A with little b in the offspring. So for a simple organism like this that has just 2 different kinds of chromosomes, there's 4 different kinds of gametes we could get, right? We could get big A with big B and little a with little b, or we could get big A with little b and little a with big B. For any organism, to figure out how many combinations there are, you take 2 raised to the n power. So if we had 3 different chromosomes, it would be 2 to the 3rd, or 2 times 2 times 2. So there would be 8 different gametes we could make. For humans, since we have 23 different chromosomes, that means there's more than 8 million possible gametes that could be produced from one individual, assuming that the homologous chromosomes had just 1 mutation or allelic difference between them. Crossing over accomplishes the same thing for genes that are on the same chromosome. So we've already talked about crossing over. It happens during prophase of meiosis 1. So if we started with all the big alleles being on a single chromosome, if crossing over happened here on the ends, now we have this new unique recombinant chromosome that has some of the original genes from this parent, but 1 gene from the other parent. So the same thing over here. We have big D, but we have the little f on this chromosome. And again, this is going to produce gametes that have a different genetic makeup than the gametes that came together to form this particular parent. Remember, crossing over only occurs during meiosis, not during mitosis, because it happens when the homologous chromosomes are paired up. So these new chromosomes that are produced that have a combination of mom's DNA and dad's DNA on a single chromatid are called recombinant chromosomes. So in this case, the ones on the outside edge have not changed. They have the parental combination of genes. But the ones in the middle where crossing over happened have a new combination. Those are the recombinant chromosomes. And we're going to come back to those in chapter 15 as well. The last thing that contributes to genetic variation is the fact that fertilization is random. So if we complete meiosis in this organism, okay, and then do meiosis 2, okay, it's possible, let's say this is a flower, something that's self-fertile, right? It's possible that we could get this gamete with this gamete, and we'd get an offspring that has a new combination of phenotypes. If we took this gamete and this gamete, we'd end up with an offspring that looks identical to the parents we started with. Or we could take this gamete and end up with offspring that have a different combination of genes. So when you put together independent assortment and random fertilization, you can get over 70 trillion different combinations from 2 parents that are heterozygous at 1 locus on every set of chromosomes for humans. So genetic variation is vastly increased by these components of meiosis and sexual reproduction.